Digestive activities in the small intestine are regulated by two important hormones. These hormones control the secretion of a lot of substances from the accessory organs in addition to other functions. These two hormones are secretin and cholecystokinin, or CCK. They have a lot of functions in common, but they also have a couple of important differences. Let's start by looking at secretin. Secretin is released by the duodenum in response to low pH. So as the stomach begins to squirt chyme into the duodenum, the low pH of that chyme is detected by the duodenum and causes the release of secretin. Secretin is a hormone, so it will be released into the bloodstream to travel throughout the body. Secretin has three important functions that I want you to know about. The first function is to slow down stomach activity. Secretin goes to the stomach and suppresses the release of gastrin so that we get less secretion and less movement in the stomach. If you remember when we talked about the intestinal phase of stomach activity, hormones, including secretin, from the duodenum go to the stomach to slow it down. The second function of secretin is to stimulate the release of bile. Bile, which is made in the liver and stored in the gallbladder, is released into the duodenum. Bile is important for emulsifying fats. That means helping fats break up into tiny little droplets, because fat doesn't want to interact well with water on its own. When you emulsify it, you break the fat into tiny droplets that will stay suspended in the water. This is important not only for the movement of fats through the digestive tract, but also for the accessibility of enzymes. Once the fats are broken up into tiny droplets, then the enzymes can go to work on them. The third function of secretin is that it stimulates the pancreas to release bicarbonate. Bicarbonate is something we've talked about before, being in the blood, but the pancreas stores bicarbonate, and it releases it into the duodenum when secretin is present. Remember that secretin is present when the pH is low, and what does bicarbonate do? It's a buffer. It can neutralize the low pH. So when the pH in the duodenum goes down, we release secretin. Secretin causes the pancreas to release bicarbonate. That bicarbonate helps bring the pH of the intestine back up. The pH of the stomach is about 1 or 2. Optimum pH in the small intestine is between 3 and 5. So we do need to raise the pH compared to where the stomach is. CCK, the other hormone released by the duodenum, is released in response to peptides and fats found in the duodenum. So as chyme is being squirted into the duodenum, the peptides and the fats cause the release of CCK. CCK has a number of functions in common with secretin. CCK also slows down stomach motility and secretion. CCK also increases the release of bile, so you get more bile from the gallbladder to help emulsify fats in the small intestine. And it also stimulates the pancreas. But this is where we see an important difference. Secretin stimulated the pancreas to release bicarbonate to neutralize acid. CCK stimulates different cells in the pancreas that release enzymes, and these enzymes are important for chemical digestion, especially the digestion of the fats and proteins that were detected in the duodenum that caused the release of CCK in the first place. I want to take a minute to talk a little bit more about the pancreas, because the pancreas is a very important organ that's sometimes overlooked a little bit because it's not actually a part of the digestive tract. The pancreas is a gland, and it's a gland that has both an exocrine function and an endocrine function. Remember that endocrine organs release their secretion into the bloodstream, whereas exocrine organs release theirs through a duct onto a surface or into an opening. The pancreas does both. Let's start by taking a closer look at the endocrine function of the pancreas. Small clusters of cells on the pancreas, called the pancreatic islets, release hormones that are important for regulating the level of glucose in the body. You've probably heard about these hormones before. They are insulin and glucagon. The pancreas releases insulin when blood sugar levels get too high. So you eat a meal, you absorb all of those sugars into your bloodstream, and it makes your blood glucose level go up. When the pancreas detects that high blood glucose level, it releases insulin. Insulin goes to the cells in the body and tells those cells to take in and to use and to store glucose for later. So as all of your cells are using up glucose, that makes the level of glucose in the blood go down. If it's been longer after a meal, that level of glucose will continue to go down until it actually gets too low. Glucose that's too low is not a good idea because then you don't have enough energy for your brain and muscles to work. So when the glucose level gets too low, the pancreas releases glucagon. Glucagon goes out into the body and it stimulates cells to begin to break down glycogen to release glucose. It also stimulates cells to use alternative energy sources. So some of your cells, we talked about the heart cells, are able to use proteins or fats. And so glucagon released by the pancreas encourages those cells to use other energy sources to leave the glucose for places like your brain that can't use other energy sources. Glucagon also goes to the liver and stimulates the liver to carry out gluconeogenesis. That means making new glucose. Your liver has the ability to make glucose from other molecules in your body. It's not always a great idea because it actually takes more energy to make the glucose than you get when you break it down for energy. But if you're low on glucose and you need to keep your brain working, it's worth it to use some extra energy to make sure you have enough glucose. So the endocrine function of the pancreas is to release insulin and glucagon to regulate your blood glucose level. The exocrine function of the pancreas is the one that has to do with digestion. The pancreas makes a large number of digestive enzymes that are released through a duct into the duodenum, and these help break down all the different types of molecules found in our food. The pancreas releases a couple of types of enzymes that are important for breaking down proteins. They are trypsinogen and chymotrypsinogen. Trypsinogen and chymotrypsinogen are very similar to pepsinogen that's found in the stomach. So remember, pepsinogen is an inactive enzyme found in the stomach. It gets activated to pepsin. Pepsin takes large proteins and chops them up into smaller peptides, smaller strings of amino acids. Trypsinogen and chymotrypsinogen in the small intestine get converted into trypsin and chymotrypsin that do the same thing. They take the little protein parts left behind by pepsin and chop them up a little bit further. So you have short little strings of amino acids. The pancreas also releases pancreatic amylase. Remember what an amylase does? Breaks down carbohydrates into sugars. And so the pancreatic amylase can continue breaking down any carbohydrates left in the small intestine into smaller sugars. There's also a pancreatic lipase. This is the main lipase in adults, and the lipase is going to break down triglycerides, fats, into fatty acids. Finally, there are also nucleases that are released by the pancreas. Nucleases break down nucleic acid, RNA and DNA. So, if we consider all of the enzymes released by the pancreas, we can break down proteins, carbohydrates, fats, and nucleic acids, the four basic groups of organic molecules that would be in the food that we eat. That makes the pancreas pretty vital to chemical digestion in the small intestine. So all those pancreatic enzymes end up in the duodenum, and so they're doing chemical digestion in the small intestine. The small intestine also has a few enzymes of its own. These are called brush border enzymes. And they're called brush border enzymes because these are enzymes that are found in the brush border. They're actually found along the membrane, the folds of microvilli on the surface of the cells lining the small intestine. Since these brush border enzymes are membrane bound, they're actually found attached to the membrane of the epithelial cells inside the small intestine. They can only catalyze reactions right there at the surface of the cells. There are three different brush border enzymes that I want you to know about. The first are a group of enzymes called the peptidases. Peptidases are important because they complete the process of digesting proteins. So if you remember what was happening with proteins, we start with a great big protein, it unfolds in the stomach, and then pepsin comes in and chops it up into smaller pieces. Then trypsin and chymotrypsin from the pancreas chop them up into even smaller pieces. These little pieces then bump up against the peptidases inside the small intestine, and the peptidases break off one individual amino acid at a time. So it takes several steps to go from a whole protein to individual amino acids, and the peptidases catalyze that last step of breaking off the individual amino acids. The other two enzymes I want you to know about are important for finishing the digestion of carbohydrates. These are sucrase and lactase. Sucrase is an enzyme that breaks down sucrose, which is a disaccharide, meaning two sugars hooked together. Sucrose is made of one glucose connected to one fructose. Sucrase is the enzyme that breaks that apart so that we have a separate glucose and fructose that can be easily absorbed. Lactase breaks down another disaccharide. Lactase breaks down lactose, which is formed as a disaccharide of one glucose and one galactose. Lactase breaks that apart so you have a separate glucose and galactose, which again can be easily absorbed. People who are lactose intolerant lack the enzyme lactase. They don't have lactase. Without lactase, they can't break down lactose. If you can't break down lactose, you can't absorb it in the small intestine. Instead, that lactose goes all the way down to the large intestine. In the large intestine, we have normal flora that can break down lactose. But unfortunately, when they break down the lactose, it produces side products like gases and acids that tend to make someone very uncomfortable when they consume dairy if they're lactose intolerant. And with that, we've completed the process of chemical digestion.